I think there's just a lot to be said for being able to integrate specific data sets around specific patient populations. The first question isn't what's the machine learning model? The first question is what are the data sets in which the most informative kinds of information are generated in the least dangerously biased way?
Welcome to Data and Biotech, a podcast from Cordi where we explore how companies leverage data to drive innovation in life sciences. Every 2 weeks, we sit down with an expert from the world of biotechnology to understand how they're using data science to solve technical challenges, streamline operations, and further innovation in their business. Here we go.
Brant Peterson, welcome to the Data and Biotech podcast.
Thank you.
Awesome. Well, just to kick us off, would you mind giving us an introduction to your background and what brought you to Valo Health?
Sure. Yeah. So I sort of went through the list of all the things you can do basic science on, right? I worked on worms and flies and mice, PhD and postdoc mostly, somewhere around information encoding in biological systems. So my training is basically in genetics and molecular biology.
Yeah. So you've got this genetic background. Can you talk a little bit about how your background in genetics and your experience at Novartis, sort of framed your approach to drug discovery?
Yeah, absolutely. So I think, when you're trained in genetics, even sort of from the very beginning, right? I think I did a fruit fly lab in high school. It's sort of distinctive from the rest of the bioeducation in that you start at the end, right? You're thinking about how this, complex causal chain that you have no hope of getting your arms around um, ends up in an organismal phenotype. And so then the other most important thing is the phenotype. And you pretty much immediately learn that's the hard part, right? Like you're sorting flies, you're not sorting them on genomes, right? Like you're sitting there looking at how many bristles there are on their shoulders. Um, and I think that sort of painfully directly translates into the really hard parts about biomedical research, right? Like, and rightfully so, the things we care about are what a patient experiences in the course of health and disease, right? We run our clinical trials on how a patient feels, functions, or survives, right? Like we have to start at the end. And I think that's actually really helpful when you're thinking through some of the really hard steps in drug discovery. Like, do we go ahead here? Do we need more data here? Do we know where we're taking this medication? Are we ready with this molecule? These are all questions that are fundamentally informed by whether or not we're ready for where this is going to end. And then I think the other piece that sort of ended up being maybe more relevant than I thought it would be at the outset is I worked a lot in grad school, especially on the mechanisms that underlie molecular evolution in traits between closely related pairs of species. And that evolution is primarily non-coding, right? So most of what that mechanistically is based on is variation in the non-coding genome and the regulatory genome. It just turns out didn't have to be this way, although I think, population geneticists would argue that in fact selection means it has to be this way, that those are a lot of the same mechanistic processes that underlie variation in health and disease in people that we see when we do these very large scale genetics studies. So I think it's, the mindset, yes, but also maybe some of the praxis turns out to be really directly translatable.
Awesome. Can you talk a little bit about like your journey with Valo Health and how what you just said is embodied in the way that Valo approaches drug
Yeah,
discovery?
absolutely. I mean, the pitch was more or less start with the end. Like that was almost it was almost as simple as that. I think I started here just over 6 years ago, and when I interviewed, it was a fairly small place, right? I think fewer than 40 employees, um, and very much still figuring out exactly how the business would work. But the notion at the core of it was that there was something about the combination of the sort of advanced computing that we can do and the data that we had access to that would let us de-risk much earlier the kinds of things that kill projects in drug discovery, right? So I had this experience at Novartis, like really rewarding experience actually, building machine learning models that predicted safety outcomes. And that was super exciting. And we learned it was going to be complicated to figure out how to get that into production in a sense, right? For good reasons, for all the right reasons. Smart people doing their jobs well in a structure that didn't give us a path. And Valo immediately, just at the outset, was built to make sure that we were pulling that information as early as we could, that the integration of kinds of evidence with the points at which that evidence could have the greatest impact would be globally optimized rather than locally optimized so that we would always be thinking about where we're headed.
Would love to hear from you, sort of like from the highest level view, like what does the data ecosystem look like that sort of feeds that on a tactical level?
I love this phrase data ecosystem. I think this is actually one of the really early things we figured out. But I just to rewind, I think your question is the answer to the question, right? So we are looking for the data that will at the earliest point, allow us to de-risk the core hypotheses that are going to impact the readout in the clinic, right? We're thinking about what's a phase 1b, what's a phase 2 readout, who are these patients, and how do we find them before we ever make a molecule so that we can understand what's going to drive the disease state, what's going to, restore health in that patient population in ways that matter to patients in clinical development. So that's the problem statement. The data flow directly from that, right? If we want to understand causality in patients, we need data like from patients, right? So where is that, right? And we can talk a lot more about the details, but fundamentally, we're looking for um, the kinds of data that give us points of leverage over understanding causality in the patient populations that we intend to treat, and understanding how to bring causal perturbations we may be excited about to the right patient population. So I think there's a little bit of a joint optimization there. Sometimes you don't like start with the patient, like have zero ideas in the universe before you go look at the patient data. It is often the case that you're working with smart, motivated biologists with deep knowledge of a particular biological process and lots of really solid ideas for how we could modulate that process. And then the task becomes, how do we find where exactly in health and disease the ability to perturb that process is going to matter the most? How do we prove it? How do we get it to patients? So that's like, that's this sort of concept of, we can notice patterns in patients, we can resolve causal structures in those patterns, and then we can use it a few different ways, sort of different but similar ways. And then I think that, the other major fork of the work is a bit more closely related to that kind of like side project I described earlier. There's a whole sort of ecosystem here of, we call it closed loop discovery or closed loop chemistry, but doing essentially that same thing with the molecule, right? So starting where we mean to end, modeling the properties that are going to make a difference to our ability to get to the clinic, like not just does it bind, right? Like, can we develop a particular hit into something that we can synthesize at scale that is going to be soluble, that is going to be safe, that's going to have good ADME properties, right? So these are the kinds of things that we can model on data before we have a hit, and then continue to refine those models as we move through the drug discovery process. So analogous to having a starting point before we get started and continuing to refine those models in the patient world, we have the same sort of mindset and process in the compound world. And then you asked, like, what's the infrastructure? What's the data architecture? Um, I think this is like, again, one of those like things we kind of came in the door with was, we're going to use the same machine learning models that allow us to predict whether or not a small molecule is going to bind a particular protein. We're going to use those models for drug discovery, for asking, does this series look like it's got a good future for modulating this target? We're also going to take those exact same machine learning models and we're going to apply them in the clinic, in not even in the clinic, in practice, in the real world, to the likely off target, undocumented off target properties of already approved medicines that patients are taking out there in the world.
So as I understand it, like the clinical data that we're talking about is coming from electronic health records or EHRs. Is that correct, or is there other clinical data going into it? And I'm interested in like how that feeds how that feeds the discovery process for you.
Yeah, I think vast, 99 plus percent portion of the data that we're going to talk about by pure volume in any given context will be real world, will be EHR. I think there's just a lot to be said for being able to integrate specific data sets around specific patient populations. So I think about, even I think frankly, like a lot has been derived from research in UK Biobank, right? Like we obviously want to have that as part of the story. But I think, specific cases, I think about an ALS, right? This is a relatively rare disease, participation of patients and their families in these data collection efforts specifically targeted around what matters in this disease is huge. So even if it's only a few thousand people compared to millions and millions of people and, tens of millions of patient years, like these numbers look big, but it's really important to plan for where these relatively smaller data sets can have outsized impact in specific questions. So that's, that aside, yes, the primary data source we work with as we're setting up our sort of core research, as we're building machine learning models, these are electronic health records. These are, real world data um, from the patient populations that we care about, but drawn from larger healthcare systems for the most part.
And my understanding is that you're sort of the EHR data that you're working with. One of the things that like is unique about how you're using it is that you're not sort of aggregating the data and attempting to make predictions in the aggregate, but that you're developing models that are sort of making predictions on an individual level based on features of that individual from the longitudinal course of their EHR records. Am I understanding that correctly, or like how does that work on your end?
Yeah, that's right. So, and I think this is, again, in many ways driven by the specific questions we're asking. So sometimes we're asking these very wide aperture questions, right? We're trying to understand the sort of disease course in very large patient populations, or sometimes even the course of many uh, call it separate diseases. They're not really, right? Multimorbidity and the progression through different symptomatic presentation or chains of drivers, right? I think we think a lot about like metabolic disease and the consequences of metabolic disease. If we think about, heart failure and kidney failure and which causes which, right? These are, you have to take a broad aperture on these questions. So in these cases, yeah, we're looking over decades. We're looking over hundreds of thousands of patients. We're looking over as much of the health information as we can pull in. Frankly, we're looking over tens of thousands of features. In other cases, we might have a pretty specific question. We might have a known off target of a drug that is of interest, and we'll be looking at sort of a target trial emulation approach where we know exactly what we need to know. We know what the clinical study would look like, and we're really laser focused on getting those data as good as we can.
Right. So there's this element of yes, you're creating sort of the general infrastructure to process large quantities of these of these like longitudinal EHR records at the patient level for it, for the purpose of training larger models, but also you're creating the infrastructure that you need to ask those more targeted questions when you have a hypothesis that you want to sort of test and understand, based on the data you have available. Am I thinking about that right?
Yeah, that's absolutely right, and that's as much a technology problem as it is kind of a culture and process problem. So a lot of what we built up is really just a like an institutional knowledge base, frankly, of how to start asking one of these questions, and then how do we know whether the question will benefit from a decade of information prior to a diagnosis, or a follow-up period longer than the baseline period, right? These are the sorts of things where knowing how to ask these questions and extract early answers quickly is something that we've built a good amount of technology to support. But the use of the technology requires that sort of understanding of the question in order to translate. So a lot of it's been building that sort of like hybrid kind of engineer scientist. I mean, 100%. So, I mean, I think the thing we inherit from epidemiology most strongly is the drive to understand the generative process for the data before we start asking questions, right? So one of the kinds of pieces of culture or institutional knowledge is understanding who do we need to talk to in order to take that step from the data as we've sort of structured and curated it in a general way to the data set that's going to best inform the next project we're going to do at whatever scale, right? Is it going to be a language model trained over tens of millions of visits, or is it going to be some very specific analysis in 5,000 very carefully selected people? The first question isn't what's the machine learning model. The first question is, what are the data sets in which the most informative kinds of information are generated in the least dangerously biased way, right? And this means we need to understand bias inherent in the generation of the data from the healthcare process, from the coding process, right? From how we see it. So that sort of upfront, sort of almost like don't touch the keyboard yet activity. We've learned a lot about how to do well, and we've learned some things that don't work as well. I think one of the things that I'll just say you have to talk to experts, right? If you're talking about data generation, you have to talk to clinicians. If you're talking about the sort of mechanisms you want to extract from the data, you have to talk to your biologist, right? It's fundamentally like a extraction elicitation of that knowledge, followed by encoding that knowledge into our analyses that keeps us from kind of riding off the rails immediately.
Yeah, no, that's really interesting, and also sort of flies in the face of a lot of what people want to say about. Just as an example, like can you just give an idea of like the scale of the
Yeah.
patient population data that you're working with, and then maybe your thoughts on like how you think about the process you just described relative to the throw a transformer at it, let it extract all the features approach.
Yeah, I mean, the first thing I did when I walked through the door here was, like, got into one of these data environments, and set up the biggest skepgram model I could, and, it ran for weeks, and it sure did make clusters, and, we got to the end and I started showing them to people, and, there were 2 really unfortunate things about that experiment. One was that the UMAP projection, looked like really unfortunately like the chocolate ice cream emoji that we use for other things, which was, just the chance happenstance at UMAP. But the other was that it was shockingly uninformative. Um, right? It told us that old people see the doctor more often than young people. And it taught us that there are a whole category of diagnostic codes and medications and even procedures that only occur in women between an average of 20 and 35 years of age. And then, you go unpack that and you're like, oh, they have babies. So like this was like a really fantastic early wake up call. I'm glad it was early. We went back to actually that exact same architecture, in the context again of these, this big, um, kind of broad sweeping question about what is true, across cardiovascular and metabolic and renal disease, what's shared, what's, not shared, what defines trajectories. And it took us several months, prior to even starting to structure the input data for PyTorch, just to figure out which people not to use, what stratifications to impose initially, which features were and weren't informative, even in how we included them in the model, right? What is a patient level feature versus what is a visit level feature required really sitting down and thinking about what is actually time varying, what matters, what do we take out, what's confounding, and I think we learned that set of lessons, and now it's almost like baked into how you do this stuff, right? We know we're going to see correlates of socioeconomic deprivation. We know they're going to show up in different ways. We kind of have an idea for what the hallmarks are, and we have a toolkit for sort of removing those axes of variance from the procedure, informed both by what we see in the data and also by what we know will be there from talking to folks who see a given patient population.
That's really interesting. And just to clarify, with like, I mean, you all are of a relatively large size, and you're talking about millions of patients over decades of.
Yeah, depending on the data set.
Okay.
So in some cases, we're looking at a couple of million people over an average of 20 years. In other cases, we might be looking at more like 8 to 12 million people, but where we have a shorter, period of good solid data from those populations on those scales.
But even at that scale, what I'm hearing is that sort of embedding your prior understanding of what are the confounders and what are the mechanisms into not just the model or not just the model you're using, but the data set that the data set that you're presenting to that model, and setting up the model for success is sort of like a key learning from early on in this phase. Is that right?
Yeah, I think the other thing is really thinking hard about what we're after here, right? Like again, like start with the end. It is easy to learn about patterns of care from data collected largely to track patterns of care. Um, it's comparatively more difficult to learn about fundamental biology from these data. I think it's not something that most folks that walk in the door here have done before. It's not sort of the most common thing that a health outcomes person would have worked on. If you worked on mechanisms of disease as a biologist, you probably didn't spend a lot of time with healthcare data. So this, like, this activity does require us to tune in some specific ways how we ask the questions, right? Like, how do we remove from the data, like we talked about socioeconomic deprivation, how do we remove from the data patterns that are reflective of care when we're not looking for care? Or how do we make sure that when we're asking about specific patterns of care, we're asking in ways that aren't sort of trivial, decedents of the disease we're asking about in the first place, right? Like these kinds of threats to inference around triviality matter a lot when what we're after is mechanisms.
Yeah, that makes a lot of sense. So, I want to talk about sort of like two aspects of your data ecosystem that we haven't spent as much time on yet. So, there's the wet lab component that you mentioned a little bit earlier, and then there's Valo also has really deep roots, and you have deep roots in genetics. So, I'm
Yeah,
interested in sort of like how the wet lab feedback loop works, and how the genetic component also enters the picture.
the answer is in different places. So the I guess I'll start with the how we think about the integration with the wet lab, because I think this was again extremely straightforward when the basis for discovery was the models that we had in the lab. When the basis for discovery is patient data, the activity of bridging becomes you know meaningfully more difficult, less obvious, I guess I'll say. And we found a couple of places, some of them I think anticipated, some of them a bit surprising, where the ability to sort of weave these two together at specific points in drug discovery has really made a difference. So I think, when we're thinking about out of all the genes in the genome or out of hundreds of potential genes in a pathway, how do we prioritize biological hypotheses? It's relatively hard actually to integrate the wet lab at that scale in these, differentiated human-centric organoid models. That the match isn't ideal. But as soon as we step into a specific hypothesis, right? As soon as we have an idea about a pathway, as soon as we have an idea about a target or a process, we've found surprisingly often somewhere in the data in the real world, there are proxies for that process. And often those proxies are either drugs specifically modulating that process or known off targets, right? Well, I guess when I talk about genetics, I'll talk about pleiotropy, but I guess I'll surface it here. Drugs are pleiotropic as well, right? Drugs impact multiple mechanisms through known and unknown off targets. We have the ability to discover some of those through that chemistry platform I mentioned, but a lot of it's just in the literature. And we've been able to use that as a pivot point um, for specific molecular investigation into non obvious pathways. As soon as we're there, the ability to calibrate what those drugs are doing in specific cells, in specific disease relevant circumstances, becomes massively enabling for those real world evidence studies. So we immediately, as we're sort of building up the DAG of the hypothesis, right, the graph that says, if you twiddle this gene in this way, if it goes up, if it goes down, we expect these markers to change in a cell. We expect these pathophysiological processes to change in a patient. We expect these outcomes to be impacted on an 18-month time frame. That's a hypothesis. Some of those arrows early on, we're not going to get in a person, maybe ever, certainly not until we go to the clinic and collect specific data, but they're cheap in a cell, right? So that ability to formulate the hypothesis and then inform different elements of the hypothesis with different pieces of evidence has been really valuable in thinking about like, which of these potential real world evidence studies are going to get us closest to the mechanism we're after. That may not be obvious in what is known in the world, that may not be obvious in the patient data, but we may be able to really incisively separate those when we go to look in the right cell type in vitro.
Yeah, that's interesting, and it also, I mean, it sort of tees us up for the discussion of genetic causal models and pleiotropy, and I would love to open that up. But just first, what I'm understanding is that the causal DAG that you're creating, the DAG for directed acyclic graph that says, these genes impact these mechanisms, which impact these mechanisms, which impact these mechanisms, which impact, ultimately the therapeutic outcome that we do or do not want to create. Having that written down is not just useful for sort of for testing the entire DAG itself, but it's also useful for identifying sort of opportunities to gather wet lab data that validate or invalidate any sort of leg of that DAG, like the relationship between one point and another. Am I understanding that correctly?
Yeah, that's right. And it's just often the case that these kind of like endophenotypic measures, right? Like what's going on kind of below the surface, right, in the tissue in the cell, are really hard to get in a patient and really easy to get in the lab. And then vice versa, right? Like we cannot ask what will the patient outcome be in cell culture. And so like this, that just is translation, right? It's almost trivial to say. But this structure lets us separate out elements of the hypothesis that we can test, that and more than test, that we can calibrate, right? That we can benchmark and put quantitative values to, that allow us to estimate if we can get 50% inhibition, we see a twofold change in this or a fourfold change in this biomarker. We can monitor that biomarker in plasma, say, right? And we can say, this is how much of that change we need to see in order to see a 13% reduction in risk of this event on a 2-year time frame. And that's our registration endpoint for the CVOT. You know what I mean?
Yeah, that's interesting, and that it strikes me that there's a lot of what would we have to believe is true in order to believe this
Uh-huh.
entire mechanism, and
Mhm.
then targeted experiments that can
That's right.
help along the way. And some of those what I'm hearing is that some of those experiments are actually wet lab experiments, but sometimes you determine you can ask that question of the EHR data with previous drugs that have been brought to market that may modulate like a similar mechanism. And so there's ways to sort of like collate this information in order to like weigh the evidence across all of the methods you have of getting at what the truth is.
That's right. That's right. And then, I mean, I think, we've alluded to it a couple of times. Genetics gives us another sort of tool in this toolkit, right? So if we think about like the sort of the canonical intersection between genetics and drug discovery, right, is this idea that we can track variation in populations in the genome to associate that variation with variation in clinical outcomes, right? So we can say, get all the people that have ever had a heart attack and get all the people that have never had a heart attack, despite living long enough to be about the same age as the people that have had a heart attack, and sequence them at, 10 million odd variants in the genome that we, know how to measure. Um, and then just ask one by one, hey, are you associated with heart? How about you? How about you? Right? Do it 10 million times and we'll come back with a list of votes, right? Um, and then we have an infrastructure built over decades and many hundreds of people's work for essentially attributing the variation in the genome that is observed to be associated with variation in health outcomes to probable mechanisms that we could imagine intervening on for drug discovery. There are lots of ways to do this. I think a reasonable approach that is fairly systematically applicable is to use the genetics of variation in those perturbable mechanisms and match it to the genetics of variation in outcomes. And this turns into sort of the instrumental variables, Mendelian randomization story that I think a lot of folks are familiar with, where we say we have the genetics of outcomes, health and disease outcomes. We have the genetics of something intermediate, the level of a protein, the level of a transcript, the level of some biomarker like, CRP or something like that, something that gives us a hint about what's going on inside the box. And we can sort of combine those two data sets to say if a particular variant in the genome impacts that endophenotype gene expression or the level of a biomarker or something, and also disease outcomes, well, we're reasonably certain CRP doesn't change the genome. We're reasonably certain, heart failure doesn't change the genome, and we're reasonably certain, that we can at least direct the arrow between the two, between the gene and the disease, say. Then we can start to ask this sort of causal question, like canonically kind of backdoor style, Judea Pearl, causal DAG type question. And when you start to do this, you're already back to building DAGs. So this has a really natural extension once you move beyond sort of the simple pairwise case to similarly populate our confidence and help us parameterize the magnitude of effect on those edges on that hypothesis that we're expressing.
So it makes sense that you're constructing this DAG, that you're using sort of the correlations between the nodes and the DAG with each other and with the outcomes to like construct this causal story, and then you're bringing information to bear to either support or refute that causal story. But there's assumptions that are built into that kind of model. So
Yes.
we'd just be interested in understanding for you, like, what are some of the key assumptions that you're testing
Yeah.
along the way?
Yeah, absolutely. So, I mean, I think we were just talking about genetics, right? And we were talking about, I think, um, an approach that has seen enough popularity that I no longer feel like I need to try to say the word Mendelian randomization or even spell it, right? Like I just say MR in meetings and expect people to know what that means. I think when something reaches that kind of sort of penetration in the field, um, it is because we know how to use it safely. And so I think this is one where the assumptions are very clearly stated, right? The approach itself, right? Instrumental variables analysis, it's like almost 100 years old. I think the first person that used the term in print is Philip Wright, who is Sewell Wright's dad, right? Sewell Wright, like founder of population genetics. So like this is like OG math, and it does a nice job of stating that one of the things that can't happen is there can't be a way to get from the instrument, the genetic variant, to the outcome any other way than through the cause. And that's not something the method proves. That's something the method assumes. And when you're dealing with genomes, biology in general, the idea that there's like only one path to anything is fundamentally bankrupt. So as soon as you're looking at what does this variant do, the answer is likely a lot. And so that a lot is this concept of pleiotropy we mentioned, right? Like the idea, I think, from kind of an organismal biology evolution perspective, is that a single gene can influence many outcomes, right? Like canonical case, right? Like alleles predisposing to sickle cell anemia are also protective against malaria. So this produces this sort of canonical balancing selection situation because those two things are inseparable. Like evolution cannot help us out with sickle cell without making us at higher risk for malaria. So in the case of this kind of like causal reasoning, the idea that a single gene could act on multiple traits gets translated down to a single variant could act on multiple genes. You could have an apparent action on multiple genes through the intervention of linkage disequilibrium, where, and again, I think we're going to end up with like a pretty big term sheet on this, but linkage disequilibrium being the idea that variants that are near each other in the genome co-segregate across populations. So it might not be actually that the causal variant for this gene is also the causal variant for this gene. They might just be close to each other. They might be the same, right? So there's some really fantastic, like delightful examples in literature. I think maybe my favorite two are the endothelin 1 factor 1 locus in blood pressure, is this case of a single region in a bit of chromatin that is accessible in endothelial cells that clearly has an association with blood pressure. And I think, you can read a lot of papers. I think very probably has that association through both endothelin 1 and factor 1 action, possibly in different tissues, right? Factor 1 maybe a smooth muscle effect. So there's this like really nice story that absolutely flies in the face of all of these core assumptions of these methods. And it's not that you don't see a signal when these things happen. It's just that you cannot interpret the signal on its own. And so this gets back to, I think, the sort of the word picture you're painting of you have to enumerate the other paths in the graph even if we have tricks, like we like the instrumental variables approach, especially when there are multiple instruments, because it lets us do a trick in the regression that essentially helps us feel better about the risk of pleiotropy. So we do this Egger test, and it's real. It's telling you something, but it's not a substitute for understanding the causal structure you're imposing. Right. The other example I really like is this obesity association with the gene FTO. FTO is in the brain, probably doing something. I mean, like there's a whole literature on how that could work. But there's also this really beautiful paper from 2015 showing that that enhancer is in physical contact with a locus containing two transcription factors that are responsible for browning in adipose tissue that's directly relevant for energy expenditure and metabolism. So it's just, I think, again, you can debate, but you have to understand that the mechanism could easily be yes, right? We may not be looking for the gene. We're looking for how this locus is assembled to causally impact by multiple paths, pleiotropic paths, this one outcome.
From a drug discovery perspective, like what does that imply about what you're looking for in the data in the DAG in order to, know that you're onto something?
I think again, this is that gene versus the gene, like if, maybe one of the genes in the locus that could be associated as a structural protein is a transcription factor expressed in embryonic development, is that, all these things that aren't drug targets, right? So we haven't talked about this so much yet, but I've sort of sketched around a few ways of generating causal evidence in humans. I said I like genetics. I've admitted to being a geneticist, whether that's a good thing or a bad thing, right? I've talked a lot about start at the end and the phenotype and all that. I even said like a good thing about a genome for the purposes of causal inference is that it doesn't change. That actually makes it a terrible proxy for drug discovery, right? Like in drug discovery, what we're going to do is make an acute intervention. Like in the Judea-Pearl causal reasoning sense, we're going to have a do intervention that wasn't present at some point, and then a patient's going to start taking a drug, and that will induce some change. So the genome cannot model this, right? It's a lifelong exposure. So when we start to talk about this, like we need to be thinking that there are obviously true causal signals that are not vulnerable to therapeutic modulation. And maybe it's not that they're not theoretically modifiable, but we also have to think practically about what drug discovery is going to look like, right? So we could have an example of something where we see that when we look at the regulation of a particular gene in a particular tissue, the direction of effect with respect to disease through these causal models goes in one way. We look in another tissue, it goes the opposite way. We could envision a therapeutic that was tissue specific. Maybe there's some antibody drug conjugate approach, but these are hard, right? Like they're just hard. So when we're thinking about prioritization of therapeutic hypotheses, I think we have to be reasoning about the sort of advanceability of the program that we would envision launching based on this resulting DAG. And it may be that when we look at the evidence, we see a really robust association with something that happens, in embryonic development or with a structural protein or something like that. And then a quantitatively weaker observed variation association with outcome in something that we think we could drug. And then, for instance, we might look at the data and see that we observe more constraint in one or the other of these, and that may tell us something. We see more, sort of tissue specificity versus broad tissue expression, right? Like a lot of other factors go into thinking about what's going to make medicine. And ultimately, I think the decision to prioritize targets is, something we spend a lot of time, frankly, on is building systems for extracting this sort of integration of evidence downstream of all of this causal reasoning and causal DAG, kind of like building the biology gets us to kind of the first checkbox on drug discovery.
Right. So you build the biology, you go through all of this work, you create the mechanism, you validate the mechanism, but you're also you have limited resources and time, and so you're trying to devote your time and attention to the places where you actually think the mechanism if if valid can be
Yeah,
drugged,
as a
can't
path
be
forward.
has a path forward. And so there might be many biological discoveries that you could make that would be interesting in and of themselves, but could not lead to the end goal of what you're investing to do. And so that's interesting in and of itself. It makes a lot of sense.
A lot of the architecture of how we implement these things in projects has to do with what we can do systematically versus what has a, sort of a higher individual unit cost around a target. So there's, in some cases, sort of trivial-ish scalable activities, right? Like when we talk about running Mendelian randomization, we know how to do that. You know what I mean? When we talk about differential expression in tissues or even sequencing patient genomes, which we haven't talked too much about, like one of the, I think, really exciting things that we're up to most of the time is identifying the most interesting patients in the real world cohorts that we have access to and pairing those with biobank sample collection to get genome sequences, to get, multiomics or at least proteomics from those patients, usually from peripheral tissue or from peripheral fluids. Those all paint fairly straightforward pictures for how to do the analysis and how to interpret any given result. Some of the other stuff we've been talking about, right? Like a lot of this sort of real world evidence, causal reasoning, target trial emulation, these are very difficult to scale. So I think there is like there's an art to when specifically in a drug discovery project, each of these tools can enter the toolkit, specifically to avoid that sort of like, everything about a gene and it doesn't help you make a medicine.
Yeah, no, that's interesting. And we've talked in sort of general terms about how you're doing drug discovery with the phenotype in mind. I would love to focus on sort of the example of diseases like Parkinson's and Alzheimer's. And if
Mm.
you're able, I would love to just hear the story of how the methods that you've shared and the data that you've gathered are applied to study diseases like those.
I guess my sort of personal interest, I'll admit it. The reason I wanted to start at Valo was because I thought there were a couple of these diseases in Parkinson's sort of front of the line, where the variation between people was the dominant characteristic of the disease, right? And there's this sort of throwaway quote, like if you talk to folks in the Michael J. Fox Foundation, you'll find this quote: "You've met one Parkinson's patient, you've met one Parkinson's patient." Right? I think there's a lot of diseases for which that's true, but Parkinson's especially, the way the disease progresses is so variable. We have these sort of incredibly poignant patient days, right? Where folks would come in and three or four people would come in and talk about their experience of the disease. And they were just completely different, like what this person's biggest challenge is. This second person looks genuinely surprised, like I hadn't thought of that. And then you talk about like, what are you worried about? What scares you? And across the board, the answer is, I don't want to lose my edge. I'm worried about cognition. But the disease, progression in the disease, UPDRS part 3, what we measure in the clinic when we do a clinical trial, motor symptoms, right? So it's almost a different disease that folks are talking about, worrying about versus what, we're measuring when we do clinical trials. So it just felt like this has to be something we approach from the level of individual patients. This has to be something we first break apart in the experience of patients in the real world before we ever start thinking about medicine. So one of the first approaches we took was that plus, right? Why not include Alzheimer's disease, right? Why not include MS, right? Like, why not understand this full sort of neurodegenerative spectrum, neuroinflammatory spectrum? We have so much evidence from the genetics of the diseases from, the 2010s to really understand that there are overlapping drivers, right? Some of the same genes are coming up in all the canonical hit lists. Alzheimer's had just been, I think, for me, fundamentally recast as a neuroinflammatory disease, no matter how hard we try to knock down, A beta or tau. So putting these patients all together and asking what differs about these patients and what's similar, we just immediately saw, like a really obvious constellation of dementia-related symptoms that sort of co-clusters Alzheimer's patients and not all, but some Parkinson's disease patients, right? As you'd expect. We saw subsets of Parkinson's disease patients that just really clearly lit up kind of an inflammatory profile. We saw subsets of Parkinson's disease patients that looked very obviously sort of mitochondrial, metabolic, just in what we saw. So you could say, oh, Parkinson's, the lysosome, the mitochondrion, but it's not everything in everyone, right? And you see it immediately in how it breaks out across patients. And then we've had some, I think, preliminary success even in trying to track those subgroups to progression patterns. We've been able to validate some of that in the sense that we saw some of the earliest onset patients were some of the highest risk for rapid progression, where that pattern's totally flipped in multiple sclerosis, right? It's the late onset patients that progress the fastest there. So this is like like a really good sort of orientation into the disease. I think where we are now is we've really focused in on a few of those patient subgroups. And I think I can't go into too deep of detail there, but to understand that these are the unmet need, right? These are the patients that are experiencing progression despite medication. Our best efforts do not meet the needs of these patients. And those are where we're starting to map out how do those patients progress, right? And we're seeing that some of those subgroups really do have that sort of canonical motor progression, and that leads us to sort of one path to the clinic for any mechanistic intervention we find there. We're starting to look at, the genomes of patients, the sort of proteomes of patients from those particular subgroups. And these become hooks for discovery there. I guess on the other hand, I'll say we've also found some really unusual kinds of disease. And I don't know that we'll be following up on them from a molecular perspective, right, from a target ID perspective. But one of the things I did not expect was that we also see a really clearly discrete subgroup of Parkinson's patients that have non-temporally overlapping lifetime comorbidity with neuropsychiatric disease. And when you really go digging in literature, there's a couple of papers that are suggestive of this, but I think that wasn't something I went in with. But I think it immediately implies maybe not so much like a drug discovery paradigm, although there could be shared mechanisms that you'd be led to if you thought about how those diseases interacted, but maybe more like this helps us appreciate a particular population of Parkinson's disease patients that have in their deep past a fairly stigmatized medical history that defines a prodrome. We have this sort of canonical constipation prodrome, loss of smell prodrome. I think there is for a subset of patients a neuropsychiatric prodrome that's just not as well understood that could really meaningfully enter into how we think about care for those patients.
Thank you for the explanations and for the stories. I mean, what it really highlights for me, based on what you're saying, is like you're talking about you're going in and you're exploring. At first, you expand the lens of what it means to study a disease, and you understand sort of the landscape of this constellation of neurodegenerative diseases, and then you understand you break it apart and you understand, what are the different manifestations of each of these diseases, and who are the subpopulations of patients that are, experiencing those different constellations of symptoms, and then, choosing, as I understand it, subpopulations that have particular mechanisms that are, potentially both understandable and potentially druggable for like further exploration. So, at the very beginning, you're understanding not just what the mechanism is, but who you might target and what the biomarkers are,
That's
and
right.
like the entire
That's right.
ecosystem. Am I thinking
And
about
that
that
takes
right?
us back to kind of one of the things you said earlier. Sometimes it'll be that this is a group of patients that we have to find a path forward for. These are the folks that nothing's working. Sometimes it's more like if I scan across this disease with a mechanism that I have a strong reason to believe is associated somewhere, where does it best sit? So, if I'm thinking about um, oxidative stress, mitochondrial mechanism, who am I looking for? And the answer is it's not everybody. There are biases with respect to age of onset. There are biases with respect to sort of male, female. There are presentation differences. The constellation of specific progression to date in the disease will help you see. And then there are biomarkers that you can measure below that, right? So I think it's um, we think of it as a map, right? It's a way into the right neighborhood. And then turning that constellation of observations into a causal pattern through a lot of what we were talking about earlier starts to help you not just bring the hypotheses you have, but generate new ones. I think that's right.
Yeah, no, that's really interesting, and I love how sort of like the framework that you've developed allows you to ask, the entire landscape of questions that you would want to ask, and attempt to ask them earlier in the process. Are there any applications of LLMs or AI that are really exciting from your perspective? Ways that it's being used internally
Yeah,
that you feel like are helpful, or maybe ways where you feel like it's not quite doing everything that that its build is
yeah,
doing. Just interested in your takes on
it's
it.
a fair question. So I mean, I think one version of the answer is that we've been training language models for over half a decade here. This is trivially true in the sense that the deep learning architectures that we've had some of the most success with on the really large scale data, right? When we're taking a quarter of a million people for 2 decades, 60 million visits, 40,000 features per patient. Like when we're taking these enormous data sets and trying to learn structure from this fundamentally longitudinally sparse, fundamentally conceptually sparse data, language is also fundamentally conceptually sparse, fundamentally longitudinally sparse, fundamentally non-real, non-matrix like. So it is an infuriating data type just like EHR. So like, when we think about the modeling architectures that make sense, um, the sort of NLP deep learning of the 20 teens, early 2020s is a really appealing match. I think we haven't so much seen a ton of success with direct token transformers. I think maybe that might be because we've got the wrong definition of tokens. So I think that it's intuitive and natural to call the medical code the token, but it isn't the order of medical codes in the record, not even temporally, that encodes meaning. I think when we think about meaning in patient journey, that meaning is much more like the episodes of care themselves. So I think we're tokenizing the wrong thing in the trivial transformer architecture, and in these sort of older skipgram architectures, we've had a little bit more freedom to find the right level of meaning on which to encode the language like concept. So I think this is just a direction for I think us as a species to work harder on. We have some research directions here. We're sort of thinking about how to get that closer to the mark. But I think that the goal remains the same. This extraction of a dense, real valued numerical representation of synthetic concepts built up from and faithful to the input data, but tuned by whatever additional kinds of tasks you want to put on the model architecture in whatever end-to-end training you want to build. It is a remarkably flexible, remarkably powerful way to take a ton of patient data and turn it into something that's amenable to sort of more standard machine learning pipelines. So from a language model inspired approach, I think this is a real like backbone kind of workhorse of what we've done over the last like half decade. From a like language model as chatbots perspective, I will say, I think if we're like appropriately skeptical, and kind of keep our heads on straight, there is a bunch of exciting stuff that kind of actually does work. So I mean, a trivial one is I just don't write a lot of plotting code myself anymore. Like I just say I'd like the points to be green, please. I think it's even more true when we're thinking about like visualizing graphs. I find network X is not a trivial syntax to write, and it is really easy to write in Cloud Code, right? So that's like these are simple things. I think there are some more interesting ones. So I mentioned things that don't scale well in our causal reasoning approaches, especially in the real world, include these really painstaking kind of trench by trench real world evidence target trial emulation studies. We have some early results that I think are really promising around um, sort of early study design details from general descriptions of hypotheses. And this is not, I think, if you take a macro lens on the industry, at all surprising, given where I think a lot of pharma companies have seen success with AI and development is in protocol development, right? So I think writing these protocol documents is like, it's not even a cottage industry, it's an industry in the AI world. And I think we're seeing like a little slice of that as valuable here. So it's again early days. I think really important to keep our metrics in front of us when we're doing this sort of thing. Really important to be interrogating reasoning traces, et cetera. But I think where we have these kind of fledgling agentic systems for performing specific tasks, there really is an opportunity for those to change the scaling logic, change the scaling economics of some of these kinds of analyses.
Yeah, that makes sense. And also to leverage the expertise you have in house more effectively because you have brilliant scientists spending less time writing visualization code and more
100%.
time reviewing protocols that are being spit out and editing them
That's right.
appropriately. That makes a lot of sense. Brant, you've been a great guest. Thank you so much for joining today. Before I let you go, can you let everyone know where they can go to learn more about you and your work at Valo Health?
Yeah, so I mean, we've got a website, valohealth.com. I think that's a really good place to get started.
Awesome. Well, Brant, thanks again for joining. I really appreciate the time and look forward to connecting down the line.
Thanks.
And that's it for this episode of Data in Biotech. If you enjoyed the episode, please subscribe, rate, or leave a review in your podcast platform of choice. See you next time.